Hey there, everyone. Welcome back to a new chapter in our study of biology. This chapter is going to focus on cellular respiration, and what we're going to encounter here is we're going to see a lot of the principles and concepts that we introduced in chapter 6 being directly applied to really what we're going to find is one of the most important processes that happen inside of cells. You can look at the topics checklist here and see that we are splitting up this chapter into 7 different videos, which may seem like a lot, but you're going to find that this is a very important process that we're going to want to make sure that we take our time with this and have good and full discussions on each one of these particular topics that you see here. This first video is going to focus on energy in living systems. We're basically just going to remind ourselves of a lot of the things that we talked about in Chapter 6, if it's been a little while since you reviewed those things on your own. So, we've looked at the net reaction scheme for cellular respiration before. You start with a 6-carbon glucose molecule and you are essentially combusting it in 6 equivalents of oxygen to yield 6 equivalents of carbon dioxide, 6 equivalents of water, and then because this is an endergonic process on the whole, an exergonic process on the whole, you're going to release a lot of energy, and the whole purpose of what the cell is trying to accomplish here is we are trying to harness as much of that energy as we possibly can and package it up into the chemical bonds of ATP. Understanding, of course, we are not going to do that completely efficiently. We're going to lose some of that energy as heat, as we have seen before. So, what you're seeing here, if you look at this reaction, that is being depicted as one chemical reaction, and that is actually not what happens in cellular respiration. That is the net reaction scheme. So basically, if you were to add up all the chemical reactions that happen during cellular respiration and cancel out everything that is in the reactant side of one process and then in the product side of another, this is what you would end up with. So this is a simple way of representing cellular respiration. But the truth is actually that cellular respiration is a combination of multiple complex reactions that are all going to occur either simultaneously with one another or in most cases sequentially, meaning they happen one right after another. So the first part of cellular respiration that we're going to talk about, called glycolysis, is in and of itself 10 sequential chemical reactions. So if that gives you any kind of idea of just how many different chemical reactions are going on here, just want to make sure that you understand right off the bat, cellular respiration is not just boom, it's one chemical reaction and then it's done. Quite the opposite. There are many different chemical reactions all happening one right after another. And we're going to see that a lot of these reactions are called redox reactions, something that we introduced all the way back in Chapter 2. If you don't remember that, that's fine. We're going to review exactly what the term redox means here. A redox reaction is going to be one that involves the complete transfer of electrons from one molecule to another or from one species to another. So the idea here is that in cellular respiration, we are going to see high energy electrons being carried from one molecule to another. So in this particular instance, if electron carrier 1, whatever it may be, transfers electrons over to electron carrier 2, that could potentially be a way of transferring energy from one place to another in the cellular respiration process. There is some important terminology that we're going to use here. So when electron carrier number 1 loses its electrons, we say that it has become oxidized. Oxidation is the process of losing electrons. When the electrons get transferred over to electron carrier number 2, we would say that that electron carrier has been reduced. Reduction is the process of gaining electrons. Now, in addition to that, you may also encounter the terms reducing agent and oxidizing agent, and this is where things can get a little confusing and a little hard to keep track of, but whichever molecule is becoming oxidized, it acts as the reducing agent. So electron carrier 1 is reducing electron carrier 2 because of that transfer of electrons. So even though carrier number 1 becomes oxidized, it acts as the reducing agent. And then the opposite is true for electron carrier number 2. It becomes reduced, but it is the oxidizing agent. it oxidizes electron carrier number 1 because it is taking its electrons from it. So if we come back and we look at the overall reaction scheme of cellular respiration, again, keep in mind this is not all one chemical reaction, but if you had to represent it as one chemical reaction, the molecules that are going to become oxidized here is glucose, which becomes oxidized into carbon dioxide, and oxygen is going to become reduced to form a water molecule. So you can kind of see what's happening there in terms of the net reaction scheme, of course understanding that the entirety of cellular respiration is actually just going to be a whole bunch of redox reactions happening one right after another, but the left side of this equation represents where we begin, and the right side is going to represent where we end up eventually. So in between here, this arrow, it's going to involve a lot of electrons kind of hopping from molecule to molecule. The electrons are going to start here on glucose, and they're going to end up here on carbon dioxide, and then the electrons are going to start here on glucose, and they're eventually going to end up on oxygen, which is going to get transformed into a water molecule. So glucose becomes oxidized, and oxygen becomes reduced into water. So like I said, the idea here is that all the different reactions of cellular respiration that we're going to be talking about are gradually going to transfer electrons from glucose. Glucose, of course, being a high energy molecule, so we're going to harvest a lot of the energy out of glucose in the form of high energy electrons. And once we leach out as much energy of those electrons as we possibly can, the low energy electrons are going to get accepted by oxygen, which will become reduced into a water molecule. Now, if you actually look at this reaction scheme here and you think to yourself, well, I'm having a little bit of a hard time figuring out what's becoming oxidized and what's becoming reduced because I can't see the transfer of electrons. If that's what you're thinking, you are totally justified in feeling that way. I totally understand. It's a little hard to appreciate here what's becoming oxidized and what's becoming reduced since you cannot actually see the flow of electrons. So to help you with that, I'm going to give you a little hint here. Oxidation very often is going to involve the removal of hydrogen atoms. So if you want to see what's becoming oxidized here, glucose is becoming oxidized into carbon dioxide. Well, what's the main thing that carbon dioxide here does not have that glucose does? Hydrogen atoms. So that's the telltale sign that glucose has become oxidized because it has lost all those hydrogen atoms, but it still has a carbon and a couple of oxygens left over. And then for reduction, this will often involve the addition of hydrogen atoms. As we see oxygen being reduced into water, you can see the water molecule has hydrogen atoms, whereas the oxygen does not. So clearly this process has added hydrogens onto oxygen to form a water molecule. So that may be a kind of a method you may want to take advantage of to kind of help you keep track of what is being oxidized and what is being reduced. Okay, so let's talk just a little bit about these electron carriers. We talked a little bit about these in chapter 6, the previous chapter that we went through. We talked about high energy molecules, and of course we are familiar with ATP, which stores a lot of energy in its chemical bonds. Now when it comes to electron carriers like NADH, FADH2, and NADPH, the high energy is not being stored in chemical bonds. It's actually in the form of high energy electrons that are just kind of lightly associated with these high energy molecules. And the benefit of this is that transferring energy in the form of high energy electrons allows us to, as efficiently as we possibly can, transfer energy from molecule to molecule without losing too much energy as heat. We are going to lose some energy as heat, but we can kind of minimize this by transferring high energy electrons rather than storing energy in a chemical bond and then breaking that bond and then reforming a bond and then breaking the bond. That is not a very efficient way of doing things, so this is why we are using electron carriers here. So a lot of the electron shuttling molecules that we're going to see here, including both NADH and FADH2, are derived from vitamin B compounds. So one of them, NADH, is derived from niacin, and then the other one, FADH2, is derived from riboflavin. Those are both vitamin B compounds. So again, a shining example of why you need to obtain your vitamins through your intake of food. So if we, in particular, focus in on NAD here, we have two different forms. We have NAD plus and NADH. So these are both the same molecule, but we are looking at them in an oxidized state and a reduced state. So NAD plus is the oxidized form that has low potential energy because it is not currently carrying high energy electrons. When NAD plus becomes reduced to form NADH, now it is carrying high energy electrons up here, and it has a great deal of potential energy. So one of the first things that you may want to focus on here before you go any further in the chapter is make sure that you kind of get it in your mind that NADH means high energy, NAD plus means low energy. So that'll kind of help you to keep track as we go into the different steps of cellular respiration. That'll help you keep track of kind of where the energy currently is. So, like I said, you'll want to make sure that you also understand that these high energy molecules are not carrying their energy tied up in covalent chemical bonds. Rather, the energy is in the electrons that are being carried by the molecule itself. Okay, and what we've kind of convinced ourselves of before, back in chapter 6, is that ultimately the goal of cellular respiration is to make as much ATP as possible. Now, NADH and FADH2 are going to be basically a means to an end for eventually making all of that ATP. And as we make our way through this chapter, we are going to learn all about how that happens. But before we go any further, just a couple of reminders about ATP, again, if it's been a little while since you covered the energy stuff that we covered back in chapter 6. So, number one, ATP is a high-energy nucleotide derivative, and we've seen before that the greatest amount of energy that it carries is in the bond that attaches the gamma phosphate to the beta phosphate group. So, if we want to harvest energy out of ATP, we only need to break this phosphodiester bond right here, release that energy, and put it to good work through energy coupling. Now, when we do that, we are going to create a low-energy ADP molecule, and if we want to resynthesize ATP, we need to reattach that third gamma phosphate onto the ADP to reform ATP. ATP allows cells to safely store energy on a very short-term basis without causing a drastic increase in free energy and heat in the cell. That would be a disaster if we were not storing up our energy for later use. We've seen before that, okay, ATP, it's not a good long-term energy storage solution, but it is some type of energy storage that we can make use of. If we didn't have it, the inside of cells would just constantly be getting really, really, really hot because of the constant release of free energy. So if that were the case, that would cause proteins and other cell components to denature because of the high temperature. And we saw in the previous chapter, in particular with proteins, you do not want the temperature to get too high because enzymes stop working very well. And then, as I was saying before, ATP is in a constant cycle of being synthesized from ADP by attaching a phosphate to it through a phosphorylation process, and then being hydrolyzed to harvest its energy through the dephosphorylation process. So the process of using an enzyme to attach a phosphate to ADP, we call that phosphorylation, and then taking it off to harvest the energy, that is dephosphorylation. So when we're making ATP during cellular respiration, we have to consider the cell has a finite, limited supply, but a constant supply of ATP and ADP if you pool them together and consider them the same thing. So if you add up all the ATP molecules plus all the AD molecules that a cell has on hand, that's going to pretty much be a constant number. So how much ATP, the high energy form, that a cell has on hand is very much going to depend on how much ADP is actively being phosphorylated via cellular respiration. So that's kind of the trick of what we're going to do in cellular respiration. We're going to harvest energy out of glucose into high energy electrons that get carried by NADH and FADH2, and then we're slowly going to use that high amount of energy to attach phosphates onto ADP. So the whole point here is that if we are not performing cellular respiration and we are not attaching phosphates onto ADP, we're eventually going to get to the point where 100% of the cell's supply is in the ADP form, the low energy form, and 0% is in the ATP high energy form. So if no phosphorylation events occur, then all the cell is going to have on hand is ADP, the low energy form. So at that point, the cell will not be able to pay any of its energy costs for its endergonic reactions through the reaction coupling that we talked about in the previous chapter. So that should convince us that our goal in cellular respiration is to harvest energy out of the food that we eat and use that energy to attach phosphates onto ADP through the phosphorylation process. So during cellular respiration, we are going to see that there are two different types of phosphorylation that we can perform, and we're going to utilize both of them. The first one is called substrate level phosphorylation. We'll talk a little bit more about this on the next slide, but basically this is the attachment of phosphates onto ADP by enzymes that get going on very early in the cellular respiration process, during glycolysis and the Krebs cycle. The second type is called oxidative phosphorylation, and this is going to be where we really start to harvest that high energy that we get from those electron carriers and use that to synthesize ATP in the mitochondria through what's called the electron transport chain. We are going to eventually see that the vast majority of the ATP that we make during cellular respiration is made by doing oxidative phosphorylation rather than substrate level. Okay, so here is a depiction of substrate level phosphorylation. This is going to be a process that produces kind of a very modest and minor amount of ATP, nothing worth shouting about, but you still make a little bit of ATP, and every ATP molecule that we make will end up counting. So during some of the reactions that occur early on in both glycolysis and the Krebs cycle, we are going to be using enzymes to make transformations of the glucose and other metabolites that we're going to be working with. And every now and then you will see an enzyme pluck a phosphate off of one of those metabolites and stick that phosphate onto ADP to make ATP. It's kind of a happenstance kind of thing. It's just like as long as we're attaching this phosphate, or as long as we're removing this phosphate from this bisphosphoglycerate molecule, we might as well stick it onto ATP and make something worthwhile. So it's not a very efficient way of making ATP, but it's still a good way of making a small amount of ATP. You might as well make some ATP if you can. But like we said, the vast majority of ATP that we're going to end up making is via oxidative phosphorylation. Now, oxidative phosphorylation can only occur inside the mitochondria, whereas substrate level can occur both in the cytoplasm and inside the mitochondria. So glycolysis takes place in the cytoplasm, and the Krebs cycle does take place in the mitochondria, but with oxidative phosphorylation, it's a mitochondria-only event. So we'll talk about this in more detail later, but what we're going to see is that those high energy electrons that we transfer onto NADH and FADH2, those electrons get transferred over via redox reactions to proteins in the mitochondria, and that energy that's tied up in those electrons is going to be used to power an active transport process. So we talked about active transport back in chapter 5, I think it was. So the thing we are going to actively transport are hydrogen ions. So we're going to actively transport hydrogen ions from the mitochondrial matrix. That's what you see, the kind of inner portion there on the inside of the inner membrane here. And we're going to pump that into the space between the outer membrane and the inner membrane, what we call the intermembrane space. So by doing that, we are going to create a huge concentration gradient of hydrogen ions, and there's going to be an enzyme that we use later on called ATP synthase, which is going to utilize the release of potential energy that comes from hydrogen moving back across the membrane in order to manufacture ATP. So as hydrogen moves down its concentration gradient, we use that release of free energy to stick phosphates onto ADP to make ATP. And as I mentioned before, this is going to be where we see the vast majority of ATP being made. So just to sum this up before we move on to the next portion of this chapter, these are the 4 phases of cellular respiration, and we're going to have pretty much a video dedicated to each. The next video that we're going to have is going to cover glycolysis. Glycolysis is a series of 10 sequential reactions, each one of which is catalyzed by a different enzyme, and these all occur in the cytoplasm. So you don't need the mitochondria in order to perform glycolysis, because this all occurs outside the mitochondria. We are going to produce a very small amount of ATP. We're only going to produce ATP by substrate level phosphorylation here. So we don't need any oxygen, we don't need any mitochondria. All we need are the glycolytic enzymes, which exist floating around in the cytosol. And the product here is that if we start with a glucose molecule, which has 6 carbons, we are going to end up with 2 molecules of pyruvate, each one having 3 carbons. So it's not quite so simple, but if it helps you kind of get in the right frame of mind, you could think of it as taking a 6-carbon glucose molecule and splitting it in half into forming 2 different 3-carbon molecules. That's not the whole story, but that's essentially the idea of what we're going to work with. Number 2 is called the prep reaction. We'll cover this in the video after glycolysis. Those 2 molecules of pyruvate that we made at the end of glycolysis are going to be oxidized and transported into the mitochondrial matrix, because from here on out, every part of cellular respiration will occur inside of the mitochondria. During the prep reaction, we will make a molecule of NADH and 2 molecules of carbon dioxide, which we form because we're going to slowly start removing carbon from those 2 pyruvates. The Krebs cycle, which we will also cover in the same video as the prep reaction, and the Krebs cycle is sometimes also called the citric acid cycle or the tricarboxylic acid cycle, so whichever one you see, just know that we're talking about the same thing here. And this is going to occur in the mitochondrial matrix, and it will produce NADH and FADH2 in the form of high-energy electrons. So we are going to further remove carbon from pyruvate and produce more carbon dioxide. So that should help to kind of answer the question of where is that carbon dioxide coming from that we produce during cellular respiration. And then finally, the electron transport chain, which will be its own video. We are going to take those high-energy electrons from NADH and FADH2 and start moving them onto the proteins that we find in this transport chain. These iron and sulfur-containing proteins make them able to accept those electrons. And as we said before, we're going to generate a big gradient of hydrogen ions that we use to synthesize a large amount of ATP. So once we have our low energy electrons, after we've successfully leached all the energy out of them, we are going to pass those electrons on to oxygen to form a water molecule. And here you can see kind of the whole picture of what we see here. Glycolysis happening out here in the cytoplasm. We produce pyruvate from glucose. We make an NADH molecule. Everything else from there on out is going to occur in the mitochondria, and future videos will cover everything that you see here, and we will keep track of all of the ATP accountancy that we're going to be keeping track of here. All right, that's going to do it for this first video. Thanks for your attention, and I will see you next time, and we will start talking about glycolysis.